Good morning. Good morning, everybody. Welcome to the Basic Science Plenary. I am one of the Basic Science co-chairs, and on behalf of Ralph Pantoflet and myself, Jimmy DiCiacos, I am pleased to introduce Dr. Stephen Deeks, Professor of Medicine at the University of California, San Francisco. We are honored to have Dr. Deeks today. He is a recognized expert on HIV-associated immune dysfunction and HIV persistence during ART therapy. He has published over 400 research peer-reviewed articles, editorials, and invited reviews. He has been the recipient of numerous NIH awards, including the principal investigator in the DARE Collaboratory. He was elected to the American Society for Clinical Investigation, and he also maintains a primary care clinic for HIV-infected adults. Without further ado, Dr. Deeks, we are honored to have you here.
It's an honor to be here. Vancouver is one of my favorite cities in the world. Always happy to fly up here and share my perspectives. What I'm going to do here is to provide sort of a broad overview of where we are in terms of HIV cure research. But before that, just let me just note my conflicts here. I do get some research support primarily from Merck, Gilead, and ViiV. So the goal here is for me to discuss really what I think is a question that the field has never really fully answered is, do we really need a cure? And if so, why? And I think that's an incredibly important thing for us to have a consensus on because it has a huge impact on what a cure will actually look like once it's developed. And then I'll discuss, from a big picture perspective, some of the broad ways that we're going about trying to achieve a cure or remission, and my thoughts on which are the most viable pathways. And then I think at the end, sort of discuss a little bit about how we're working to harness the immune response to achieve a long-term remission. So, really important question, and there is not a really consensus on this. Given that antiretroviral therapy is so effective, why do we actually need it? What is the unmet need that we're not actually achieving in the context of established infection with regard to treatment? And so there are two perspectives here. One is the individual perspective. I'm a clinician. When I got into this business, I thought basically the reason why I want to cure is obvious. People don't like taking pills. There's all these comorbidities. It's difficult to adhere to these drugs on a day-to-day basis. They're expensive and so forth. But it's been quite interesting, actually, in a number of surveys that have been done among people who actually have HIV, asking them a question, well, what is exactly that you want from a cure? And a very consistent finding, which I got to tell you really surprised me, is it's actually stigma. It's this concept, and I think a lot of people in this room are very aware of this issue, that living with HIV on a day-to-day basis, sure, you can retain your health and so forth, but there is this ongoing societal stigma that I think is best illustrated by whatever happens when someone in the media comes out with HIV infection and just really describe the tension that they go through. So when you ask someone with HIV why they want a cure, this is basically the number one thing. And if it's stigma, if you want to get rid of stigma, that's actually very difficult to do. That actually probably means to most people not just being off antiretroviral drugs, but actually not harboring any HIV and ultimately being HIV antibody negative. So that's what the individuals generally want, and that implies actually a true sterilizing cure, which is going to be difficult and challenging to achieve. But I think, from my perspective, the investment that's happening globally in terms of HIV cure is driven less by what the individual person with HIV desires or needs, but more from the fact that antiretroviral drugs, on a global perspective, may not be able to fully address the needs and stop the epidemic. And there is some data here. So this is from, and I think I'm not an epidemiologist, but I think most people in the room are familiar with this. Over the past 10 years or so, there's been an amazing investment on a global level for antiretroviral drugs, and an unprecedented global response has happened with drugs now being distributed worldwide, including very much in sub-Saharan Africa. But despite the fact that there's been this global investment, probably a little bit more than half, I'm sure these data are changing, of the global population have been able to access HIV antiretroviral therapy and achieve an undetectable viral load. And as a consequence of the fact that the penetration has not been complete, there is ongoing HIV transmissions, and up to a couple million last year. So treatment by itself may not necessarily be able to end the epidemic unless the global response is able to basically get the remaining 50% on antiretroviral therapy. And with waning resources and certainly in our country, less of an investment for what's happening elsewhere in the world, that may be difficult to achieve. But even if it is, and this is, again, a controversial issue. So there, in San Francisco, like in a lot of places up here in Canada, particularly in Vancouver, we've had a massive investment in the city to try to basically get to zero. And San Francisco has devoted a massive amount of resources to this question. The Department of Public Health, the academic institutions, the community-based institutions, the clinicians have all worked. We actually work quite well in San Francisco in this regard. And we have PrEP is widely available. There's signs everywhere about getting to zero. There's signs everywhere about getting people on PrEP. And we have a culture now in our city of starting everybody on drugs immediately. And in fact, I've had a handful of people who've come to my clinic who have plus or minus evidence for them being HIV infected. And our standard is make sure they leave with medications in their stomach before we even actually finish doing the workup. So we treat everybody immediately, and this is something that the entire city is bought into. So even in that setting, we have reduced transmission events probably 50%, which is great, but we are probably not necessarily going to be able to get to zero. And the reasons for that are all societal. A lot of people with HIV, for whatever reason, are not as engaged in healthcare and so forth. So treatment itself, I don't think is necessarily going to be able to stop the epidemic. From my perspective as a clinician, the big problem with treatment is not necessarily challenges getting people diagnosed, getting people on therapy. We can do that. It's keeping people on therapy. Just things get in the way, right? In our city, when Obamacare emerged, it just was a nuclear bomb in terms of how we actually deliver care. A lot of people accessed new health insurance, lost their old health insurance, big breaks in their health care. And now that Obamacare may be dismantled, we're doing the same thing. People, get in the middle of midlife, they get burnt out, midlife crisis. There's just numerous issues that actually prevent people from, once they staying on therapy indefinitely. And so I think that's actually the major unmet need with the antiretroviral drugs, the fact that people need to access them, take them on a daily basis, and do so for decades and decades. And that's not actually going to be easy for many, if not most people, to achieve. So that, for me, is the major unmet need. And that has a huge implication as a curologist, as someone involved in developing cure strategies, on what a strategy, what a cure must look like. And so that gets to this issue, another ongoing debate in the field of definitions. And I think there's now consensus on this. When we talk about cure, when we actually say cure, what we're really saying now is the complete and total removal, elimination, or silencing of all replication-competent HIV in an individual, okay? And that was what we think we need to achieve in order for someone to be no longer infected, to be antibody negative, and to reduce the stigma. A very important goal, but one that's going to be difficult to achieve. The Berlin patient, we all know about Timothy Brown and what happened to him over a decade ago. He actually moved to San Francisco a few years ago. We spent 2 years poking and prodding and trying to figure out, in fact, whether or not he actually is cured. And we found evidence of virus here and there. May have been real, may have been background noise, and so forth. So even if we think we can achieve this, a true cure, it's going to be hard to define. Proving a negative is difficult. And I'm going to show a case report about this challenge in a few slides. So this is the desire, a cure, but it's going to be difficult to achieve. Most people in this business have decided right now that the most viable pathway to achieving art-free health, right, getting people on therapy, then allowing them to stop therapy and remain free of disease, which achieve the public health desires of an intervention, that this will be more viable. And this we generally refer to as a remission. Remission is now defined as durable control of a virus that we know is there, but it's maintained indefinitely at a level that is that does not cause negative consequences to the person, and very importantly, as at a level that does not make a person likely to transmit the virus to others. And we think this is going to be possible in large part because it's been done. It happens naturally in the lead controllers. And as I'll discuss a little bit, it actually probably is now happening as a consequence of a therapy, antiretroviral drugs, in the so-called post-treatment controller phenotype. A question always comes up, though, here, going back to my hometown. We did a study several years ago where we took elite controllers, people who achieve remission, and we found out that these individuals have fair amounts of inflammation and probably excess amounts of heart disease as a consequence of the inflammation. And our standard of care has sort of changed now. We treat now most of our elite controllers. So the question is, if we're actually treating elite controllers, why do we want to go out there and develop a strategy of treated people and turn them into elite controllers? And that's a great question. And of course, two reasons for that. Number one, again, we're targeting our approaches here, not the people who can take therapy, but the people who cannot, for a variety of reasons. And there's also this really interesting subset of controllers who have been referred to as exceptional controllers or extraordinary controllers or whatever word you want to use, who actually do look quite healthy and pretty normal, have low reservoirs, no inflammation, zero risk of rebound as far as we can tell. And so those are the true models for achieving a remission. And those extraordinary controllers, again, are rare, and there's only been a handful reported in the literature, are actually quite similar to the post-treatment controllers, a point that hopefully I'll make in a few slides. Okay, so that's the background in terms of why I think we need a cure and how to define a cure. So how is this going to be achieved? There are basically 5 very basic approaches that are being pursued in the clinic, outlined here. If I was a betting man and someone wanted to bet me what is the easiest way to cure someone, I would say without question it would be using gene therapy or cell-based therapies. There are just a number of different viable pathways that this can achieve, and the reason why I think this is the most viable pathway is, of course, because it happened in the Berlin patient, and it almost happened in a bunch of other transplant settings. So this is certainly the way to go. I'm not going to spend any time discussing this in detail, in large part because what I think personally, what we need in terms of a strategy is something that will achieve that unmet need on a global level. We're going to need an intervention that is actually scalable and affordable across the world. And it's not clear to me whether or not the gene therapy approach is going to achieve that, although there are potential mechanisms for this to happen, the so-called gene therapy in a box that people are working on. A second way of achieving a cure is the one that was most popular when this whole cure business started several years ago. That's the so-called shock and kill. The approach here is pretty straightforward. We know that the major barrier to achieving a cure is the fact that the virus persists indefinitely in these long-lived resting memory T cells and perhaps macrophages and other cells, that the virus is essentially silenced or not silent or hard to detect. The immune system therefore can't get at it. So we're giving these various different interventions to shock the virus out of its hiding place so it can die, so the cell will die. Proof of concept has been achieved in people. We have shown with a number of interventions that you can actually reverse latency. You can actually get RNA production from latency. You can even get virus production at very low levels. What we have not shown in people, however, is that by doing this, the cells that you actually are reversing latency from die or anything good happens. Although there is some emerging studies from the monkey model that indeed, in fact, actually, at least in certain kind of monkeys treated very early, it is in fact possible to perhaps cure in monkeys with this shock and kill approach. Again, a topic I'll discuss at the end of my talk. But I'm of the mindset, and again, it's my opinion, that shocking and killing every last virus that exists in a person out of its hiding place and killing it is going to be a very challenging thing to do, and that shock and kill as an end, as a way to cure people, is probably not going to be feasible, but it's going to be very important to reduce the reservoir. All the models of an HIV remission start off with people having very low reservoirs, and so any approach that causes a shock effect and then removal of a big chunk of the reservoir is going to be incredibly important as an adjunct. There's another approach that hasn't actually made it into the clinic yet that's quite interesting to me. If you take these resting memory CD4 T cells from a person on long-term therapy, it's actually hard to get the virus out of these cells. It's basically formal. It seems like it's fully integrated. It's got all this sort of methylation and these genetic modifications. It's in a part of the genome that's hard to transcribe. Essentially, it's in deep latency. And it's certainly possible that there's a big chunk of the reservoir that people carry in their body, in fact, is in such a deep state of latency, for whatever reason, it will never come out. And there are a bunch of therapeutic approaches that are being pursued in the laboratory to basically do this to the entire reservoir. Starting off with these TAT inhibitors that have been shown, at least in the laboratory, now in mice, to cause permanent latency. And there are a number of pathways, including some that are now actionable. In fact, some that are being studied for other reasons in people with HIV, including blocking Mtor and some other pathways that, in fact, may be able to contribute to achieving this state of deep latency. And so this approach, called block and lock, again, is in the background now. It's not yet made into the clinic, but it's something that I think a lot of people have a lot of excitement and promise around. Early ART. All right? Can we cure people with early antiretroviral drugs? You can cure monkeys with early antiretroviral therapy, but what about people? So, in getting back to our getting to zero approach in San Francisco, again, we have all these stakeholders that have come together, and we're basically pouring antiretroviral drugs, either as PrEP or treatment, everywhere that possibly can go to get rid of every single virus in the city. This has led to a series of interesting scenarios. We now have several now, of people who are at very high risk for HIV, and there's this one case I'll talk about here, very high risk for HIV, multiple partners, wants to go on PrEP. And so we have these studies to do this. This gentleman about 2 or 3 years ago decided he wanted to go on PrEP. He came in, he screened. He was HIV, it was like on a Wednesday, he was HIV negative. And he came back on a Monday, he started PrEP. On that day, his viral load was 51 copies. So and we confirmed that it was real, and he started PrEP on that day. So he started therapy essentially on day 1 of Phoebic 1, okay? The earliest you can possibly diagnose HIV, just by luck. We found out a few days later that, in fact, he was infected, and I think within about 4 days or so, he was put on standard antiretroviral drugs. So he went from Truvada to a standard 3 or 4 drug regimen. And then for 2 years, we tried to figure out whether or not he was infected. And we did again. We lymph node biopsies, cut biopsies, he had a lumbar puncture. We leukapheresis a few times. Samples were sent all over the world to try to figure out exactly what's happening. In fact, Nicolas Chamont in Montreal was a major player in trying to figure out what was going on in this particular setting. And everything was negative. He was antibody negative. His T-cell activation was normal. He had no evidence of an HIV-specific T-cell. We could find nothing. So we actually thought he was cured, but we didn't know what to do. And the patient was very aware of what was happening, and he got fed up with all this poking and prodding and decided on his own he was going to stop therapy. And so we hurried up and wrote a protocol. He stopped therapy, and he came in basically weekly for many months. He was traveling all over the country. We set up laboratory, went to Disney World with his family. A viral load, the way to get your viral load tested right outside Disney World. I didn't know that. And it was boom, boom, boom. And it was an interesting experience, I got to tell you, because we're nervous. We were quite nervous because if the virus rebounds in this setting with no pre-existing HIV immune responses, it can be quite ugly. When we stop therapy in most people, there's a pre-existing immune response that actually blunts what happens. And so we did this for 9 months, and it's actually, he didn't know, is it going to happen today? It has an impact on, his interactions with his partner. It was a kind of a stressful situation for everybody, monitoring him. But then the day before, like a few days before Thanksgiving, he came in, he got a blood test. It was sent off. We had our 4-day Thanksgiving break that we have. It got lost in shipment and so forth. There was a bit of a delay. He came in a few days later than he should have. He looked terrible. He just looked absolutely terrible. And the viral load from a few days ago was now just barely detectable. So we were pretty convinced he was HIV infected. I was worried he was having acute retroviral syndrome. We put him on therapy at that time in the room. He left the clinic on treatment. And it turned out on that day, his viral load was several thousand, so it was real. And in hindsight, what we think happened is he had a flu or something that probably activated the virus, so he wasn't sick as a consequence of an acute retroviral syndrome, but perhaps some viral syndrome, in fact, caused the virus to rebound. So it is probably impossible to get and this is kind of similar to Mississippi Baby, that was in which treatment was started on day 1. It's probably impossible in people to truly cure people with antiretroviral drugs. There's always going to be something left that you have to worry about. And in fact, the Thai cohort, there's about where they have 10 or so people who started therapy in Thailand in FEBIC 1. They've also basically found that essentially everybody rebounds when you start therapy very, very early. A take-home message from me is that one of the most important things that we're going to do in the cure agenda is diagnostics that allow people to figure out at home or easily whether their virus is around or not. If people are on treatment, they know that their essentially virus is undetectable and they can go on their life accordingly. But when you're in a remission and there's virus around and you don't entirely know what's happening, that actually is something that we need to figure out how to address. And there is a big initiative from the NIH to develop these types of assays that will allow someone to do this type of stuff at home and so forth, which I think is going to be very important. So curing someone with treatment, 100% cure is not going to be possible. But how about achieving control, right? So there's this post-treatment controller phenomenon that everybody's aware of that occurs. It's actually been reported for about a decade now, but the landmark study that sort of put this on the map was the Visconti study. This was an initial description published a few years ago now of 14 people in France who started therapy early, but not super early. Okay, that's a really important point. Most of these post-treatment controllers are people who start therapy usually around 2 or 3 months into their infection. But there's this phenomenon where the people start therapy, they're on therapy for a long time, the longer the better, stop therapy, and anywhere from 10 to 20% of those individuals do not rebound. And that's quite phenomenal in my mind. It is pretty clear now to me, I've always been a bit dubious about this phenomenon, it's pretty clear now that if you get antiretroviral drugs at the right time, in the right kind of person, and you keep it going for long periods of time, years, maybe 1 in 10 or 1 in 5 can stop therapy safely and the virus doesn't rebound. These guys immunologically look like those exceptional leak controllers I talked about. They often have very low reservoirs, tiny reservoirs, and very low levels of immune activation. But we do not know the mechanism for control. There was an interesting randomized clinical trial that was just done, and this is way too small to see, but it's an interesting paper. The NIH, Tony Fauci and his group, did a randomized clinical trial of a vaccine versus placebo in this kind of patient population, people with early antiretroviral drugs, and stopped therapy. And there were 3 people in the placebo arm and 1 in the vaccine arm that didn't rebound, providing the best evidence so far that this phenomenon of post-treatment control does occur in 10 to 20% of people who start therapy early, stay on therapy for years, and stop. So we got to figure out why that is. So treatment is basically causing a cure. What we know a little bit about these post-treatment controllers is the following. When people who are going to be a post-treatment controller stop therapy, the time to rebound doesn't really differ that much from the people who become a post-treatment controller versus people who don't. The peak viremia, however, is lower in post-treatment controllers, but not always. Sometimes it goes up very high. So if we're trying to figure out whether someone's a post-treatment controller, we're going to have to allow people to go through a period of acute viremia, which can be quite high, and then the virus comes under control. And once that happens, my sense is about half of the people will control for several months or so and then probably rebound. But the other half just go on for years and years and years and years with extremely low reservoirs, no infectious risk, normal immune system, and so forth. So a very compelling series of stories suggesting that a remission is possible. Now, how are we going to do this, right? Early ART, great, particularly if we have a biomarker that will tell us who can stop safely. That would be wonderful. But that's not going to have any implications for 95%, probably more, of the HIV-infected world. We need a therapeutic intervention that can do this. And this is where my usual talks start, and I go on for 2 hours babbling on how we can do this. But I felt because the audience was pretty diverse in terms of cure work that I would give a big picture overview of how we might go about doing this. But there is a massive investment in this arena of China remission. It's, again, being inspired by elite controllers and post-treatment controllers. But probably more than anything, it's being inspired by advances in cancer immunotherapy. If you think about it, people with cancer have cells that are diseased but look normal, and they reside in tissues that are hard to get at. And that the best way to get rid of them is to enhance the immune response such that the immune response, primarily T cells, find those hard-to-detect disease cells in tissues and kill them. Right? That's what cancer immunotherapy is doing, and it's doing in the context of an immune system that's dysfunctional. These tumor tissues are often inflamed, which actually results in the immune system not working very well. It's the identical story in HIV cures research. And so the synergism between cancer immunotherapy and HIV cure therapy is tremendous. And in fact, if you trace back where all these immunotherapies that are revolutionizing cancer, where they came from HIV money, a lot of it, in the 1990s. All these various pathways were discovered in the context of HIV-associated research. Drugs were made. They were heading toward HIV work. The cancer stuff happened, and the entire investment shifted to cancer because it should have. That's where the unmet need is. And now some of this stuff is shifting back. But how are we going to do this? All models of HIV remission show that you have 3 things, right? You have a low burden of disease, low reservoir sizes. You have very low levels of inflammation, which I think is incredibly important in this story, and you have a response that is potent and sustained. And how is this going to be achieved? Well, we need to recognize that we've been trying to do this for 20-some years, right? Therapeutic vaccines. the therapeutic vaccine is essentially meant to enhance the capacity of the immune system to control the virus in a post-treatment interruption period. 20, 30, 40 studies have been done. All have been essentially negative, right? There's some hints that when you do therapeutic vaccines, you actually can move the dial a bit. And it's pretty consistent, but it's nowhere near remission. So these approaches have failed. And why have they failed? Well, they have failed for these reasons. Number one, the vaccines that we've used are pretty weak. The vaccines we're using in cure, as well as the vaccine we're using in prevention, are pretty weak immunogens. They don't generate the kind of responses that we need. The responses that are often generate are typically dysfunctional. They express PD-1, CTLA, and so forth. They express all these different pathways, suggesting that they're not going to work that well. This has been well established. And of course, very early in untreated infection, you develop escape mutations to these T cells in particular, as well as antibodies. They get deposited in a reservoir, they don't go away. There's this inflammatory pathways that exist in long-term treated disease. Chronic inflammation results in upregulation of all these immunosuppressive responses. 99% of what the immune system does in a chronic setting is try to dampen inflammation, and that actually can prevent T cells from working very effectively. People with established disease have high virus burdens, and there is an emerging story that the virus resides in certain sanctuaries, particularly the B cell follicles and lymph nodes, that T cells and other cells can't get to. So these are the actionable items that we need to basically address with therapy. And believe it or not, they're all being done. In fact, we are addressing each of these pathways in a variety. We've got about a dozen clinical trials in development or in the clinic or just finishing that are in isolation addressing each of these approaches. I don't have any data to share. But there is a series of monkey studies that I think are worth mentioning. Number one is this well-known work by Louis Picker, where they've taken CMV, very inflammatory virus, they re-engineered it to be an HIV/SIV vaccine. They vaccinate a bunch of monkeys. They infect the monkeys. They all become infected. But half of the monkeys with the CMV vaccine, the CMV/SIV vaccine, half the monkeys become infected and the virus replicates, a reservoir is established, and in about 2 years, it's gone. Proving that we actually can, with a very immunogenic vaccine, cure monkeys. And we're working with this group to start doing this study, looking at this in people. There's a second approach that's being taken. This is work led by Dan Baruch, again in monkeys, where they've taken monkeys, put them on antiretroviral drugs. They got a vaccine with Ad26, a very immunogenic adenovirus vaccine that delivers HIV and SIV proteins, and boosting with MVA. And what they showed in this monkey study is that if monkeys got a therapeutic vaccine and a vaccine adjuvant, GS9620, which is being developed by Gilead, if you got that combination of a vaccine adjuvant and this vaccine, the monkeys, they were not cured, but a few of them, maybe up to 30%, achieved long-term elite control with really low reservoirs. That was quite remarkable. So proof of concept in the monkeys that at least in some monkeys with a vaccine and a vaccine adjuvant, you can achieve a remission. And perhaps more exciting, this was again Dan Baruch. This is another monkey study, presented late breaker at CROI, where they took 40 monkeys again, put them on, infected them for a few days. It's kind of artificial. It's a really early art. But anyway, they infected them for a few days, put them on very potent art for 2 years, which is remarkable for a monkey study. And then they got either these broadly neutralizing antibodies that are very hot in the prevention field these days. These are antibodies that can target V3 on the envelope that's expressed in cells that are infected and producing virus. So anyway, these monkeys who got this antibody, and again, the TLR7 agonist developed by Gilead, the combination, half of those monkeys were cured, as far as we could tell, no virus left, and when the treatment was stopped, no rebound. The other half had a little bit of virus left. It rebounded, but then it became undetectable again. So essentially all the monkeys were essentially either fully cured with a very simple combination of an antibody and a vaccine adjuvant. So half of these monkeys were either cured or achieved a long-term remission, providing evidence that this actually can be achieved. And in fact, there's some evidence now in people. This is a Spanish study showing that with therapeutic vaccines, some of them, this is one that looks at a very immunogenic vaccine with a chimp adenovirus and an MVA boost. There's some early signals that are emerging in the clinical literature that we can actually move the dial in terms of achieving remission. So to conclude, just a few comments and opinions. With regard to elimination and cure, eliminating full eradication infected cells is possible with early art in monkeys, but and with bone marrow transplantation, but in people, early art's not curative and transplantation is not scalable. There is proof of concept that we can cause shock and kill. We can shock, in particular. Whether or not we can do shock and kill remains debated, but I am not a fan of this approach as an end itself, just as an adjuvant for other approaches. Block and lock, as I said, remains quite promising, but not yet in the clinic. So finally, with regard to immunotherapy, I believe, many else do, that the immunotherapy, again, because of the oncology work and our work on the controllers, may prove to be the best way to achieve a remission. There certainly is proof of concept that this happens. There are a bunch of therapeutic approaches that are in the clinic now that we can actually use, that we can borrow from the oncologist, and multiple pathways are now being pursued, and time will tell. There's probably over the next 2 or 3 years, some of these clinical trials will have a readout. And with that, I'm happy to take any questions. Thank you very much. Up here. You're very welcome. I can see him, but I lost my glasses, so I can't see anyone except in the first row. But
So you didn't speak too much about gut, leaky gut, microbes. Do you think that's part of the agenda or not?
Oh yeah, it's huge. So I think that the chronic inflammatory pathways that are due to the leaky gut, to HIV production, to co-infections like CMV, that this persists during long-term therapy, that's a consequence of this inflammatory pathway, all the immunosuppressive, so PD-1, for example, but all these various different pathways are upregulated. IDO and these myeloid suppressor dendritic cells, these are some of the more classic pathways that are upregulated to dampen the inflammation from the leaky gut, and those pathways prevent T cells from doing what they need to do. This is a basic premise in oncology now, that inflammation causes immunosuppression, and that to get the immune system to work better, you've got to block inflammation. Again, I could go on forever. Half of the clinical trials that we're doing in the clinic right now are aimed at blocking those inflammatory pathways. So I think we'll have to deal with chronic inflammation before we can cure anyone. And in fact, I think post-treatment controllers are doing so because they start early. They start therapy so early that the persistent inflammation is not a major problem. Thank you for the question. Any other? If anyone's raising their hand, I can't see you, so. But maybe in the back.
Hi, could you comment on the Mtor modulation?
Yeah, no, Mtor is an interesting pathway. If you block Mtor in mice, they live longer. It's a pathway that's associated with all sorts of key determinants of healthy aging and so forth. And so there's actually a some interest in these drugs as a way to prevent aging. And also they tend to enhance the capacity of the immune system to respond to antigens and so forth. Problem with Mtor inhibitors is that they can be quite toxic, and so they're used in transplant to block rejection and so forth. But in terms of HIV cure, we did a study where we showed that by blocking Mtor in the setting of a transplant with sirolimus, rapamycin, the reservoir went down. And we've just completed 2 clinical trials, prospective studies of looking at these rapamycin or another Mtor inhibitor to see if its impact on chronic inflammation, which we think is bad, as well as the reservoir. So that's a very active area of investigation, both in gerontology and in chronic infectious diseases. But there is this new story that's emerged suggesting that if you block this pathway, you can achieve some lock and block effect. So we're quite excited by these drugs. But everything with the immune system is always, you're always looking for 51% good and 49% bad. And there's definitely downsides of using Mtor inhibitors. It's a challenging drug. Okay, well, I think we're good. Thank you very much.
Thanks to Steve, and thanks for the previous speaker. Thank you all for attending. I invite you to go over to the poster exhibit during your break and have a look at the posters.